working here. So last time we focused on the heart. Today we'll talk about blood vessels, the roles of the different blood vessels, the structure of the different blood vessels, how we have the ability to regulate blood pressure, different strategies for that. If our blood pressure gets too high, what do we do? What do animals do? If the blood pressure gets too low, what are some of the adaptive responses? We'll conclude by talking a little bit about a physiological adaptation that affects circulation, and that is diving. So we'll talk a little bit about how animals, primarily seals, but also others, can remain submerged for a very long time. And a lot of what we've spoken about as far as the heart, and what we'll talk about today in terms of blood vessels, you'll see how that kind of comes together and how animals adapt to the changes associated with diving deep into the ocean. All right. Oops, hold on a second. Let me back up a second here. New share. Here we go. All right, let's see here. Okay. So this actually happens to be a picture that I'll refer to at the end of the lecture. It's not the most pleasant type of picture, but it was a study that was done basically forcefully submerging an animal underwater. Animal survived, but basically as a way of looking at certain physiological changes associated with diving. All right, so like I mentioned, today we're going to talk about blood vessels. Hopefully you've retained what we spoke about in terms of the heart, as far as the structure of the heart, the different circuits, right? Systemic, pulmonary, at least in mammals and birds. But then we also spoke about the different types of hearts and circulations with fish versus amphibians, such as the frog, versus crocodilian or diving reptiles, and a lot of the changes that occur with that. And again, we'll probably have a little time in the beginning of class tomorrow. Well, actually, so tomorrow we'll actually start respiratory. I know I've got it down for 3 lectures on cardiovascular. We're only going to really need 2. So tomorrow, what I might do is use that as an opportunity to kind of review the cardio, what we've done so far. All right, so vessels. So first and foremost, of course, arteries versus veins. Arteries are blood vessels, of course, that carry blood away from the heart. Veins, of course, are vessels that carry blood to the heart. Now, the type of blood that's found in these vessels will vary. If we're looking at the systemic circuit, the arteries carry oxygen-rich blood. If we're looking at the pulmonary circuit, that's where the pulmonary artery comes into play, that carries oxygen-poor blood or deoxygenated blood. Veins, if you're in the systemic circuit, they carry oxygen-poor blood, such as with the vena cava. But if you're in the pulmonary circuit, those pulmonary veins bring oxygen-rich blood back to the heart. So it's just a reversal of the nature of the blood, whether it's oxygen-rich or oxygen-poor. After the arteries leave the heart, they get smaller and change somewhat to these smaller branched vessels called arterioles. We'll talk a little bit about their structure. And the arterioles continue to branch further into smaller and smaller vessels into capillaries. And capillaries are the site for gas exchange. This is where oxygen is dumped off into organs, and carbon dioxide is removed and carried back to the heart. So this is a very important site, and you'll see why these different vessels have different functions. It all has to do with the structure of them. After the CO2 is picked up from the capillaries, the blood then passes into small veins, otherwise known as venules, and ultimately back into the veins. So those are the different vessels, and a little bit about their nature of the blood that's found within them. Now let's talk a little bit about structure. So I always like to focus first on the large vessels, and that would be the arteries versus the veins. So we're going to look at the two extremes. Now, if you were to slice into both of those, histologically, you'll find that both vessels, both arteries and veins, comprised of 3 layers of tissue. This innermost layer, and the innermost layer is what makes contact with blood, is known as the tunica interna. Some books call it the tunica intima, but tunica interna. It's a very thin, simple squamous layer. If you've taken anatomy or anything like that, histology, simple squamous is a very flat cell, kind of looks like a fried egg, with the yolk being the nucleus. Outside the tunica interna, we have this muscular layer, which is known as the tunica media. And you'll notice in both large vessels, we have both. Here's the tunica interna, tunica media, this orange, tunica media, this orange, that's smooth muscle. And then this outer connective tissue, which is the tunica externa, the outside. One additional feature I wanted to mention is that just outside the tunica interna, we have elastic tissue or elastic fibers, which is more predominant in the arteries than the veins. This gives the vessel, just by the name implies, elasticity to be able, as it stretches, to recoil, which is especially important with arteries. Now let's take a look at going from the arteries to the arterioles. First thing I want to mention with arterioles is essentially we've lost one layer. We still have the tunica interna. Now the term endothelium is the same thing as simple squamous. We have the tunica interna and we have the tunica media. We've lost the tunica externa. So we have 2 out of the 3 layers. The venial also has 2 layers, but the layer that it's lost is the tunica media. So it only has the interna and the externa. Finally, we have the capillaries, which you remember, this is the site for gas exchange, as well as, I might add, exchange of nutrients and waste. This is all that's left here is just the tunica interna, which are these just flat cells. And this provides a great surface for diffusion of material back and forth. So that's kind of a general overview. Now let's take a look kind of at a more broad perspective. Here we can see really 4 different categories. We have large vein and large artery. We'll talk about that. We're going to ignore the middle one, medium-sized vein, muscular artery. It's similar, but again, there's no need to go over that. And then we'll go over venial versus arterial and capillary. Just like I mentioned on the previous slide, both arteries and veins have 3 layers. But I do want to point out, and you can see very carefully, there are some differences between these two, arteries and veins, in terms of the thickness of a layer. In the case of the artery, the tunica media is much thicker, and it has a lot more of the elasticity, the elastic fibers. Now, structure dictates function. The blood going through the arteries is under a much higher pressure than it is in the veins, so you need that thick muscular wall to be able to withstand that pressure, and for that vessel to recoil after it expands, to be able to return to its original shape. So that's the big, excuse me, that's the big difference there. Now let's jump to venials versus arterioles. Like I mentioned, arterioles don't have a tunica externa. They just have the interna and the media. Although it's hard to tell from this picture, percentage-wise, the arterial has the highest percentage of smooth muscle of any of the vessels in the body, of any type of vessel. And the significance of that is the arterial is a vessel that is responsible for kind of regulating blood pressure, whereas an artery serves to kind of as a conduit for blood, kind of having some elasticity to it, kind of conducting blood, moving blood down. The arterial helps to regulate pressure. Is a pressure regulator, the best way to think about it. In contrast, the venule has no muscle. It's a very flimsy vessel. There's nothing to hold its shape. It's frequently collapsed. And one of the consequences of having these veins that have a smaller amount of muscle than the arteries and arterioles is the fact that it becomes difficult to move blood back to the heart, especially in the lower extremities. You don't have the muscle that's going to help push it back. But I'll explain how we're able to do that, especially when you're looking at blood returning from the lower extremities. Finally, we have the capillaries, or the blood vessels, smallest blood vessels that contain just simple squamous cells, ideal for nutrients and waste and gas diffusion. All right, so a little bit of a summary here. Arteries, and I might add arterioles, carry blood away from the heart. The large arteries have a lot of elastic fibers, which acts as a rubber band. The more you stretch that artery, the greater is going to be the drive to return it back to its original shape. So it expands with blood pressure and recoils or returns back with relaxation. As people get older, or with people who have coronary artery disease or cardiovascular disease, the elasticity, the elastic fibers break down. So the ability to return back to its original shape, and this elasticity is not what it once was. And so the tendency for blood pressure to elevate and elevate uncontrollably, what we call hardening of the arteries, can occur. Arterioles, like I mentioned, have the highest percentage of smooth muscle. These are really the pressure regulators, really regulating blood flow to different areas of the body. The diameter of the arteriole can either dilate or constrict, depending on what's going on. For example, if you're running or exercising, the arterioles going to your muscles, skeletal muscles, will dilate. By the same token, the blood vessels going to your gut or digestive system will constrict, and vice versa, right? If you're eating a meal, the dilation will occur going to your gut. This change in diameter, due to the high percentage of smooth muscle, also regulates the blood flow going through the capillaries, which ensures that the blood is not flowing too quickly. And this is important because it allows for more of an efficient gas exchange. Sort of like a bus driving too fast to drop off or pick up passengers. The bus should slow down or stop, right? So people can come on, people can get off. All right. So I was mentioning about this whole concept of elasticity, why that's important with arteries. In this diagram, this area right here represents the heart. And if you can follow along where the dashed lines are, this is normally that would be the size of the heart. And this would be the aorta leaving it, where the dashed lines are. So where the dashed lines are, that's the diameter, that's the size before the heart contracts. When the heart contracts, it gets smaller, squeezes blood through the valve. Remember, in this case, it could be the aortic valve or the pulmonary valve. Blood goes into the artery, pushes up against the walls, stretches, right, the arterioles or the arteries. And then what happens? They recoil. And as the recoiling occurs, this allows blood to be pushed downstream. So elasticity in arteries is very important to ensure blood flow, the continuity of blood flow. All right, speaking of blood flow, this is a physiology class, so there's always some equations, and we'll get into more of these as we go along, but basically there's an equation that describes factors that can influence blood flow. How much blood is going through an area per unit time. And one factor that can affect blood flow is resistance. Essentially, blood flow is inversely proportional to resistance, right? So what does that mean? The greater the resistance on the denominator there, the smaller the flow. And this kind of makes sense. One factor that influences resistance is diameter of a blood vessel. And it makes sense that if a blood vessel is larger, there's going to be more blood going through it, there's less resistance. When that vessel constricts, there's less blood flowing through it, right? There's greater resistance. So this is really identified on this diagram. This is an example of blood flowing through a vessel. And we just randomly gave it this number, flowing through at a rate of 4 liters per minute. This blood splits off into smaller arteries and arterioles, and these arteries or arterioles have an equal diameter. So same diameter, so you can see that the flow is the same in all of them. However, if we have a situation where one vessel is constricted, this happens, the blood flow through that vessel is less, right? Greater resistance due to the constriction, less flow. But there's also a compensation. To compensate for this, these other vessels actually will take on what's left. You can see right here. They're all taking on 1 1/4. So it's diverted through those 3. So smaller diameter or smaller radius, greater resistance, less flow. Now, where the actual equation comes into play is going to be on a law that's known as Poiseuille's law. We'll take a look at that in just a second. But before we get into more discussion on regulation of flow, let's kind of travel to a couple other vessels. So we've spoken about arteries and arterioles. What about capillaries? Capillaries, of course, are the smallest blood vessel. They're 1 cell thick, right? These are the cells. These are these endothelial cells or simple squamous cells. The diameter is very small. The blood flow through this is very slow. And this is where gas exchange occurs. So those are kind of go hand in hand. Slow flow acts to increase the delivery of oxygen and to increase the uptake of carbon dioxide. Now, having said that, so this is the generalities of all capillaries, but there are differences between some. And we actually have 3 different types of capillaries. They all are 1 cell thick. They all are involved in gas exchange and exchange of other substances. They're the smallest blood vessel. The difference between these 3 has to do with the porosity of them, or the space between those cells. For example, we have what are known as continuous capillaries. This is a continuous capillary here. This is a continuous one here. This is probably a better picture. You notice these cells are just almost like glued together. There's very little space between them. I mean, the fact is there is some space, but very, very small, very little. These are called continuous capillaries because it really restricts what can go across. Gases can go across, but large molecules could not. We find this in areas such as the brain, where we have what's known as the blood-brain barrier, central nervous system. We find this also in the lungs, muscle, places like that where we have continuous capillaries. We also have what are known as fenestrated capillaries, where you can see it kind of looks like Swiss cheese. We have these intracellular pores that allow for a more wide variety of substances going across. We find this associated with the endocrine glands. Remember, endocrine glands secrete hormones, steroids, peptides, proteins, and they can go across there. This allows them to easily enter the blood or to leave the blood. The kidneys and the intestine are also an important place for fenestrated because both of those places involve a lot of filtration, right? Remember, the kidney is a very important blood filter. And also absorption of water, absorption of nutrients, which all occurs in the intestines. So a lot more substances can cross the cell layer in this case. Finally, we have what's known as discontinuous capillaries or sinusoidal. These are associated really strictly with hemopoietic organs, places where blood cells are formed, places like the bone marrow, the spleen, the liver to a lesser extent, although it is important during fetal development and in children. These are large leaky capillaries that actually cells can pass through. All right. So all three types of capillaries are similar in the sense that they're one cell thick. The difference has to do with the space between them and what can get through. All right. So I mentioned blood flows through the body, right? From the heart, it goes into the arteries, arterials, capillaries, venules, veins, and then obviously eventually back to the heart. Well, the speed of blood flow varies. It's highest where we have the large arteries. And you can see right here, if we take a look at this velocity of blood flow on the y-axis, it's greatest at the aorta, and it gradually gets less and less and less till it's slowest around the arterioles, capillaries, and venules. Now, why is this? Well, it has to do with the fact that once you go from arteries to arterioles, there's a lot of branching, right? If you think of a tree, branches into large branches, which branch into smaller branches and smaller and smaller and smaller. Just picture that in our vascular system. The greater the branching, the greater the surface area. The greater the surface area, the slower the flow. And you can see this right here. The greatest surface area is in this area right here with arterioles, capillaries, and venules. And that also happens to be the area where the lowest velocity is, which is associated where gas exchange occurs. So we want it slow. We want a more of an efficient delivery. And this number is not a random number. It is true. There's roughly about 40 billion capillaries in our bodies. It's kind of hard to fathom, but because they're microscopic. All right. So while we're on the capillaries, I want to focus a little bit about an interesting dynamic with capillaries. Remember I mentioned with capillaries, we move oxygen from the capillaries into the tissues, and we move CO2 from the tissues back to the capillaries. And the same is true with other nutrients. Nutrients goes from the capillaries to the tissues. Waste, other types of waste go from the tissues to the capillaries. How can you have this two-way street? Well, it's all governed by pressure. Pressure kind of rules the roost here. And it all has to do with the type of pressure and the amount of pressure. There's two types of pressures I want to look at, hydrostatic and osmotic. And depending on the levels of those two pressures determines where we're going to move stuff out of the capillary and where we're going to move stuff in. This capillary is divided up into a couple parts. What you see, this kind of a reddish area, is called the arterial end. This is where an arterial is coming in. This area over here is the venous end. This is where capillary will lead into a venule. At the arterial end, we have what's called a net filtration or a net flow out of the capillary into the tissue. Why is that? Because one of the pressures, the hydrostatic pressure, which really is the blood pressure, the force of blood coming into the capillary, far exceeds what's called the osmotic pressure. Now, we haven't spoken about osmosis yet. We'll talk a lot about that when talking about the kidney, but I'm sure you guys know from other classes, osmotic pressure has to do with the amount of particles. The greater amount of particles in a solution, the greater the osmotic pressure. So at the arterial end, and what's going to happen is osmotic pressure tends to draw fluid towards it to try to dilute it. Let me think of a good example. Well, say, for example, saltwater fish. We'll talk about this later as well. Saltwater fish live in an environment in which the water is saltier than the inside of their bodies, right? So there's more salt outside than inside. The osmotic pressure is greater outside, so that tends to draw water out of the fish. And what's interesting about this, even though they live in water, fish could actually become dehydrated if they didn't have any compensatory mechanisms, which is something we'll learn about that actually prevents that water loss. So if we think of osmotic pressure in the capillary, that's going to draw fluid in. But in this case, the hydrostatic pressure, which is the outward pressure from the capillaries that's pushing pressure to drive fluid out, is greater than the osmotic pressure that would draw fluid in. That's why we have filtration at the arterial end. At the venous end, as a lot of this fluid leaves, right, the hydrostatic pressure is less, and it's even less than the osmotic pressure. So at the venous end, the osmotic pressure exceeds the hydrostatic pressure. So now we're going to draw fluid in. This is a net absorption. So filtration at the arterial end, absorption at the venous end. And you'll see this on the next slide as well. This is out of your textbook. Really, the osmotic pressure itself doesn't change. It's the hydrostatic pressure. So here, hydrostatic pressure is high. At the arterial end, it's greater than the osmotic pressure. Here at the venous end, the hydrostatic pressure is less. And so that's what dictates either the drive out or the drive in. Pretty cool stuff, I think. Okay. Finally, the last vessel I want to talk about are the veins. I'm not going to say anything other than about venules. Just know that they have those two layers, tunica interna, tunica externa, no muscle. What can we say about the veins? So the veins, based on the number of veins in the body, you would think, okay, there's going to be an equal amount of arteries and veins. Blood's going to be equally distributed between both. And the fact is that's not true. Most of the blood in the body, in the circulatory system, I should say, is found within veins. And that's because they're very compliant. Because they don't have as much muscle, they can open up more. They're a bit more flimsy, become kind of like a reservoir. Relative to skeletal, or relative to arteries, they have very little muscle, very little elastin. In some cases, they don't have any. And because they're so far away from the heart, the pressure is very low in veins. One of the things, some of you may already recognize this, but we'll talk about it more as well, is the role of pressure in driving so many different mechanisms in physiology. We'll learn with the respiratory system. It's pressure that allows oxygen to go into the blood. It's pressure that allows CO2 to go back to the lungs and out. It's pressure, obviously, that forces blood out of the capillaries, or fluid out of the capillaries, right? Those things. In order for something to move, you have to have a pressure gradient, right? And whatever you're looking at, whatever parameter, is going to move in a direction from high pressure to low pressure. So the reason why blood, let's go from here. The reason why blood goes from the aorta to the arteries to the arterioles is because the pressure is actually greatest in the arteries, high pressure to low pressure. So just keep that in mind, high pressure to low pressure. Well, the veins already have very low pressure. So, and especially veins in your legs. So you're going against gravity to begin with. How do you get blood to go back to the heart? The veins themselves can't, like we mentioned, they really aren't adequately designed to do that. Well, fortunately, we do have a couple adaptations. One, as you can see right here, these valves. But let's talk about a few more. So again, this can happen in many veins, but really what I'm talking about is from the lower extremities up, because the challenge is going against gravity. Number one, we have what's called a skeletal muscle pump. Now, this is not a new structure. It's not like it's a special pump. It's just muscle that surrounds veins. And then when that muscle contracts, like it does when you're exercising especially, but even when you're not, that squeezes the veins. When it squeezes the veins, of course, there's blood in there. And as you can imagine, it could squeeze the blood up and it can also squeeze it down. Well, we have valves, these two valves. These are one-way valves. So what's to stop the blood from going down? The fact is that so if you look at this, this muscle squeezing, the blood can go down there, but it's gonna stop at that valve. It can't go any further. Whereas when this squeezes, the blood going up opens this valve. So it goes in a superior direction. So even when you're not exercising, they work, but that's why exercise is especially important, because they even help with venous return even more, moving those muscles. So that's the skeletal muscle pump. We also have what's known as the respiratory pump, which I'll show you on the next slide. That's another mechanism. But I wanted to show you the importance of these valves. We often talk about the importance of valves in the heart, but the valves in our veins is just as important, because if these valves don't open and close like they should, or they stay open all the time, like you see in this scenario with varicose veins, when the muscle squeezes the vein, the blood might go up, but it's going to come right back down. And that's why people with varicose veins have a lot of fluid buildup down in their ankles. And sometimes it's very painful. All right, what about the respiratory pump? So we haven't spoken about the respiratory system yet, but this will kind of come into play here. There's a very intimate association between those two systems. They need each other, right? Every time you breathe, and I'll just say inhale or inspire, what happens is the lungs inflate, right? The lungs inflate, you inhale. Well, the atria also get bigger. One of the things we know from a law called Boyle's law is that there's an inverse relationship between pressure and volume. So as the volume of the atria increase, so let's specifically talk about the right atria, because that's where the blood's coming into from the lower extremities. As that volume increases, that's going to drop the pressure. The pressure is already low down here, but this pressure drops. Combine that with the muscle and the valves, and that's going to help draw the blood back up to the heart. So every time you breathe, you're doing yourself a favor in, well, not only living, but moving blood back to the heart. So it's a pressure gradient. All right. A little bit more about pressure and how it applies. I mentioned a while back, several slides ago, that pressure, I should say, blood flow is inversely related to resistance. And I, at the time, mentioned that resistance, a good example of that is diameter, right? The smaller the diameter, it's going to be harder for blood to push through because there's greater resistance. Dilated, less resistance, blood's going to flow through much better. Well, we're going to take this and other parameters, put them into what's called Poiseuille's law. This was actually a law that doesn't just apply to blood vessels, but any sort of tubing or pipe engineering, things like that. But in terms of physiology, what does Poiseuille's law say? First of all, blood flow is directly proportional to the blood pressure gradient. Okay, so what do I mean by that? Well, let me just go back to a new share for a second. Let me go back to this. All right, so if we take a look, all right. All right, so say we want blood to go in this direction. Let's see if there's an arrow. Oh, cool, there it is. Blood to go that way, okay? Well, in order for blood to go in that direction, right, you're gonna have a certain pressure up here, and I'll just type this out. We'll call this P1, P1. Down on the other side, we're gonna have a pressure down here, it's P2. right? In order for this blood to go in this direction, P1, I don't think there's a greater, it's P1 is going to be, has to be greater than P2 to ensure that, let's see, right, that flow, right? If the pressures are equal, what's going to happen? You're not going to get any flow. So I'll get rid of that, right? It's going to stay static. Or whatever. So it's gonna stay static. If the pressure here, for example, with P2, if P2 is greater than P1, which you wouldn't want, if P2 is greater than P1, what's going to happen? That's going to completely mess things up. I know what I got it. Sorry about this. There you go. I'm learning as I go. It's going to go like that. You don't want that, right? So to make a long story longer, P1 needs to be greater than P2 if you're going to drive the blood in a desired direction. Okay. Okay. Second of all, blood flow is inversely proportional to resistance. We already know that. Okay, so let's take a look at this diagram and do a little analysis here. So what we know so far is that blood flow is directly proportional to delta P. Delta P is just P1 minus P2 over resistance. Okay, but let's kind of open this up a little bit. So blood flows if a pressure gradient is present. We know that. Blood flows from areas of high pressure to areas of low pressure. We know that too. Blood flow is opposed by the resistance of the system. We know that. Fourth one, we don't know that yet. And this is actually discussing about, besides the radius or the diameter, there's a couple other factors that determine resistance. One is what we call the viscosity of the blood. So if you think of viscosity, right? Viscosity, something that's very viscous would be something like honey or oil as opposed to water. The greater the viscosities, you can imagine it's harder the flow is going to be more difficult. If someone, for example, is dehydrated, maybe their concentration of blood cells are high, that's going to affect viscosity. It's going to be too high. Last one is also the length of the system. The further and further away you get from the heart, it's smaller and smaller vessels. The vessels get smaller and smaller, and that becomes, because their diameter is getting smaller, that provides a greater resistance. Okay, so that's all I really want to go over, those first four. Now let's plug this into an equation. And here's everything, and with a couple other factors as well, okay? This is Poiseuille's law. Blood flow is equal to delta P times, and this is where we've sort of added a few things, times pi always finds its way in there, times radius to the fourth power. It's going to be very significant. Over 8n, and n is just 1, 8 times the length times the viscosity. So what do we know? We know, so these three factors are resistance, right? Length, viscosity, and radius, right? The greater the length, the greater the resistance, the lower the flow. It's on the denominator. The greater the viscosity, the greater the resistance, the less the flow, right? In this case, the greater the radius, right, the less the resistance and the greater the flow. But not only that, we're looking at radius to the fourth power. So this is Poiseuille's law right here. So I want to focus on that radius to the fourth power because it really gives an indication of the significance of why it is maintaining a healthy blood vessels, right, and what happens when plaque builds up. All right. This is, as you can tell, kind of an old diagram where they're measuring in cubic centimeters per second, but let's ignore that right now and focus on the numbers. If we say, for example, the flow rate is 100, if we double the length of a vessel, we know that length, we know, why is that doing anything? We know that length is on the bottom, right? If we increase the length, we're going to decrease the flow rate, so it'll drop to 50. If we make the blood more viscous, it's also going to decrease the flow rate. This is kind of a misnomer, double the pressure. We mean double the pressure gradient, right? You know, instead of the difference, maybe the pressure P1 is 10 versus P2 is 5, maybe if P1 is now 20. So it's more of a steeper gradient, you're going to get greater flow rate. Look what happens when you double the radius. Think of that pi to the 4, or a radius to the 4th. When you double the radius, 1 to the 4th, first of all, as you know, is 1. 2 to the 4th is what? 16, right? So again, this is just an individual vessel. You're never going to see huge flow rate changes like this in the whole body, but you can see the dramatic impact radius has on flow rate. Now, if we take a look from a pathological circumstance, this is showing vessels that transition from healthy to gradual plaque buildup, as indicated by this yellow. And don't worry about the numbers, the actual numbers per se, but just look at the trend. If we go from an artery that has 0 occlusion, meaning that's very clean, we've got, this is the flow rate and this is the pressure, if the pressure is that. If the body's always trying to compensate, if we have plaque buildup, the flow rate is less, and we try to compensate by increasing the pressure. Pressure is going to go up to try to force the blood through a smaller opening. If we have even greater buildup of plaque, look at the flow rate there. I mean, obviously, this is not compatible with life right here. But the point is you can see that as much as an increase in diameter is really important, but what happens if a decrease in diameter will have a much greater impact, right? If you go from a diameter of 2 to a diameter of 1, you have 1/16 the flow rate. So it's pretty significant. Okay. All right, so moving along in a little bit more physiology, let's talk more about pressure. We've already spoken about pressure. Pressure is what drives flow. And here you can see this is in the heart. And of course, this goes up and down because of the heart contracting and relaxing. But let's kind of go from here where we're looking at the arteries. And you can see the pressure is highest in the arteries, and then it gets less in the arterioles, gets less and less in the capillaries and venules. And again, why is this? Obviously, further away from the heart, less musculature, and of course, that high pressure to low pressure is what allows blood to move continuously in this direction. All right, so you notice this term mean pressure. So what is mean pressure? Okay, well, the full name for it is mean arterial pressure. And what it is, it's a function of 2 different factors. And again, we have, in this kind of short, intense course, we can't go over everything, but mean arterial pressure is the pressure within an artery at a given time. What determines it? I like to think of the old input-output aspect. If this is our artery, right? We have an input of blood coming in the artery from the heart. We call that cardiac output. Every time the heart pumps, it's moving blood into that artery. That's going to contribute to the pressure. What's also going to contribute to the pressure is the arterioles, their diameter, right? If the diameter of the arterioles is really small, this is meant to be like a vice, constricting them, that's going to prevent blood from leaving the arteries, or at least reducing the amount, and that's going to keep the pressure built up. So what does this mean? Mean arterial pressure, this is the input, this is the output, is really a function of cardiac output. As you increase the cardiac output, you're going to increase the mean arterial pressure. As you increase the resistance, as indicated by reducing the diameter of the arterioles, you're also going to increase the pressure. Now, normally that doesn't happen in us. Usually we have a way of regulating it. So say if the heart is beating really forcefully, the arterioles will dilate. So it'll prevent the buildup of pressure. All right. So there's pressure all around us, right? But let's reference the blood pressure. As you know, we know from homeostasis, blood pressure is one of those parameters that we have to regulate. Some people, it's easier than others, right? In many of us, we can just, our body can do it. It's got a built-in system to do it. Some individuals, that system's not working properly and they require other, maybe other medical intervention that will help them with that, whether it's medication or things like that, okay? So how do we regulate blood pressure? First of all, blood pressure is monitored by specialized groups of cells called baroreceptors. If you think of barometric pressure, these are specialized groups of cells that are found in two areas of the body. They're found in the aorta, and they're found in the carotid artery, and they're called the carotid bodies and the aortic bodies. But they're basically baroreceptors. As blood flows past them, they're monitoring the pressure. Much like a thermostat in your house, right, monitors the temperature. And there's you sort of have like a level, a comfortable level, right? Well, you've got a pressure level that's designed for you. And if the pressure goes above that or below that, that is sensed by these baroreceptors. All right. So what do these baroreceptors do? So certainly they sense pressure changes, but how does this fundamentally result in changes in or adjustments? So if the pressure is too high, how do we bring it down? If the pressure is too low, how do we bring it up? Okay. Well, let's take a look at this example. This is if somebody has hypertension. And this could just be more of like a sudden hypertension. When you're looking at essential hypertension, or basically someone who has a chronic condition, this system does not always work properly. But, and there's a lot of other factors involved, but we're looking right now at the autonomic response. The blood pressure goes up. The baroreceptors fire signals, right, via sensory neurons that are going to carry the signal to the medulla oblongata. So let me go back here for a second, okay? So there's the medulla. There are these baroreceptors, okay? Now I do want to mention, so what type of sensory neurons are taking the signals back? Well, there's two. We have the glossopharyngeal nerve and also the vagus nerve. Those are two sensory nerves. Those of you that have taken anatomy, you know about those being the cranial nerves. These are two nerves, the vagus and the glossopharyngeal, that pick up the message from the carotid bodies and the aortic bodies and carry that signal to the brain, and they report it. What this does is, because the pressure is high, and if what you think of the autonomic nervous system, you think of like fight or flight, the sympathetic is typically kind of mobilizing us, increasing heart rate, increasing breathing, and parasympathetic is kind of rest and digest, slowing down. We would think that we want to respond in the parasympathetic way, right? And that's in fact what happens. The NTS is actually a region of the medulla where these neurons enter into, the cardiovascular center. So within the cardiovascular center of the medulla, the response is to activate the parasympathetic system. And what does that do? First of all, what that does, it acts on really primarily one area, and that is the heart rate. Remember we were speaking about the conduction system of the heart, the pacemakers? The parasympathetic acts on the SA node to slow down the heart rate. Slow down the heart rate, that's going to help in slowing down, reducing blood pressure. But combine that with the fact that not only is the parasympathetic going up, but the sympathetic is decreasing. Normally the sympathetic increases the force of contraction of the heart, right? Which increases pressure. The decrease in sympathetic output also acts to reduce the force of contraction. So you've got two things going on. The increase in parasympathetic is decreasing the heart rate, right? And the decrease in sympathetic is decreasing the force of contraction. You put those two together, contraction and the heart rate, that's going to collectively reduce cardiac output, which is back here. Okay, that's one factor. But there's also a vasodilation. So remember I mentioned that when this vessel is constricted, that increases the pressure. That same response to high blood pressure also causes these vessels to dilate, which allows, it's kind of like an escape valve. Right? So not only do we decrease cardiac output, we decrease the resistance. Collectively, this is going to lower the mean arterial pressure. So that's how we respond to high blood pressure. What about low blood pressure? Now, there's different causes of it. I'm going to use a good example, kind of a temporary example, what's called orthostatic hypotension. Say if you're lying in bed and your alarm goes off and you just jump up, suddenly you feel a bit lightheaded, right? Well, that's low blood pressure, right? So when you stand up and you get that effect, what happens is this slows down the firing of the baroreceptors, which has the opposite effect. This increases the sympathetic output, which is going to trigger a vasoconstriction. It's also going to increase cardiac output. So we're going to get an increase in resistance, increase in output, which is going to increase blood pressure to reestablish it. So you can see how these kind of are the yin and yang of each other, right? And that's really how our autonomic system works. It's kind of this counterbalance. All right, so to kind of finally end up this autonomic control or the discussion of control of high blood pressure, I did want to mention one other aspect, how we can regulate blood pressure. The first one I spoke about was already this one on the left, right? If blood pressure is too high, this activates the sympathetic system, which will decrease cardiac output, and by causing vasodilation will decrease resistance, which will lower blood pressure. We already know that from the previous slide. That's a very fast response. But then we have a slower response, kind of a backup. I don't want to say a backup system, it's just as important, that involves the kidneys. The blood pressure is high. One factor can be the blood volume. The fluid volume of the blood is high as well. It's putting pressure on the walls. So one factor we can do is to excrete more. And there's several different hormones that play a role in this. We don't have time to get into them now. But those of you that work in Professor Glombotsky's lab, he studied a hormone called atrial natriuretic peptide that it plays a role in that, in promoting excretion to lower blood pressure. So this is just kind of taking a look at the big picture. Now, this is actually not anything new. This is just a different way of looking at it, kind of a nice little tabular format. The different effects of the two branches of the autonomic nervous system on blood pressure, the effects on blood pressure. Parasympathetic, by decreasing heart rate, decreases blood pressure. Sympathetic, by increasing heart rate and increasing contractile strength, basically that increases cardiac output, right? to increase blood pressure. But also notice from the previous slides, the sympathetic system also causes constriction. And by causing constriction of the blood vessels, that's going to increase blood pressure as well. All right, so that's kind of the summary. Pardon me for rubbing my nose. My just allergies are going bonkers. All right. Now, after going through all this about kind of generalities about veins, arteries, pressure, blood flow, I want to give you one interesting adaptive response to a very interesting animal. A lot of interesting animals, but there's a scientist from years ago. His name is Knut Schmidt-Nielsen. That was his last name. He's a well-known comparative physiologist. Went all over the world, Antarctica, the desert, studying different environments and animals. What he said, for every system in the body, there's an appropriate animal to study that. For example, when you're looking at nerves, we know the squid axon, great animal. When you're looking at reproduction, right? Cattle, 9-month gestation. When you're looking at cardiovascular adaptations, the diving, right? Here's the seal. Now the Weddell seal is used a lot because it is one of the best divers of all the marine mammals. All right, so let's take a look at some aspects of how the Weddell seal dives. This is a chart which shows the duration of a dive in minutes, and this is what fraction of each of those dives, how many of each type of dive do we see? So as you can see right here, most of the dives, maybe up to 15 minutes, or maybe even the majority of dives are less than 20 minutes, right? Most of them are really short, like from 0 to 5 minutes. 40% of the seal's dives are less than 5 minutes. 20% are 15 minutes or less. But there's a small fraction where they can actually stay under for an hour without surfacing. Now, except for some extreme cases, I think maybe in Ripley's Believe It or Not or whatever, there might be some extreme individual that can do this, but this is virtually impossible. I mean, this is an air-breathing animal. So how can it do that? All right, well, let's take a look at some other aspect. These are not, so the last one was looking at the duration of the dive. This is looking at the depth. And again, you see right here, about 70% of the dives are less than 200 meters, which 200 meters, that's like what, 600 feet? But again, there are some that are even deeper. This is like, 1,000 feet. This is over 1,000 feet, right? This is 600, this is almost 2,000 feet right here. I mean, that's pretty impressive, right? So what sorts of strategies, I mean, does this animal utilize, there must be some physiological adaptations that allows it to do it. Well, we're not going to go over everything, but I'm going to go over some of the main cardiovascular aspects. If you look across the spectrum of different seals, and I think these are all seals, there's some sea lions, and then comparing them to humans, right? We're way at the end here. This is a graph that examines the oxygen storage capacity in our bodies. And it's divided amongst 4 different factors. Number 1, the amount of oxygen that can be stored in the lungs, the amount of oxygen that's carried in the blood. You can imagine, like, for example, hemoglobin, right? The amount of oxygen that can be stored in the muscle. Remember, with type 1 muscle, myoglobin, that's in the purple or magenta. And then the light blue, oxygen dissolved in other body fluids, other than the blood or anything else. So let's do a little comparison. Now, obviously, we're pretty low in most of this compared to the seal, but you can see we've got fair amount in the lungs, fair amount in the blood, but we don't store much in our muscle. And this bottom part is pretty much the same across all the animals. As we start, and I'm not going to go through all of these individually, but let's take a look at the walrus, right? The walrus, pretty much, it looks like a little increase in the amount that's in blood, but pretty dramatic increase in the amount that's stored in muscle. Let's jump to the Weddell seal. There's even a hooded seal, which is even better. But anyway, and you can see some of the numbers here. These seals can store roughly twice as much oxygen that we can. You take a look at, for example, the Weddell seal and some of these others, their oxygen storage capabilities are tremendous. Look, for example, here. If you take a look, maybe this is, it's hard to tell from the numbers here, but clearly the amount that's stored in the blood in us, compare that the amount that's stored in the blood in a Weddell seal, huge difference. Not only there's a lot more in the blood, but there's also a lot more, especially in the muscle, right? Really not any difference in this other column right there. It's even less in the lungs. So it seems like those two parameters, the myoglobin, right, which would be in the muscle, and the blood. So why is there so much oxygen contained within the blood? Why is there not a lot in, if you take a look, in other body tissues? All right. Well, this is, I think, is the coolest picture, and I've got some bigger ones to show you, all right? So what they did with a couple seals is they looked at seals that were breathing air, which is what normally they do, and seals that were submerged. And they injected them with a contrast dye. If you've ever had like any sort of imaging, sometimes they'll inject a contrast dye so they can see different areas of the body, whether it's your stomach, kidneys, whatever, right? The idea was to see where the dye goes, because where the dye goes correlates with where the blood's going, right? So let's take a look at the air breathers. And I think I'm going to go to here for a second. This is a little bit easier, all right? So when the seal is breathing air, oh, let me back up for a second. So let's take a look at some of the vessels we're talking about. So this is the aorta. This branched vessel, this is the celiac trunk. This is what goes to some of the digestive organs. Stomach, liver, pancreas, right? This right here is the renal artery, and as you might expect, goes to the kidney, right? All right, let's take a look. So breathing air right at the time they injected the dye, and you can see where the dye is going. It's going right through the aorta, and you can see these are the renal arteries. These are the celiac arteries, and you can see the blood is flowing through those arteries really well. Within 2 seconds, you can see a lot of the blood that's flown into that artery is less. But one thing that's really key is you can start to see the organs lighting up, especially there's your kidneys, right? The blood is definitely going into the kidneys. And it's a little fuzzy here, but this would be some of the digestive organs. This would probably the pancreas, liver would be up there. In 6 seconds, you can see even more. So we're going to take a mental snapshot of that and take a look at what happens in a diving seal. All right, within 1 second, so I don't know why they didn't do the same, but within 1 second, okay, there's the dye, it's in the aorta. You can see some of it is entered into the renal vessels, they're in the celiac vessels. So let's compare where it is 1 second here versus half a second. And you can see already the difference. Here it's already made its way to the organs. Here, it's not. And even after 4 seconds, maybe a little bit better, but still, it doesn't really go into those organs. And it's going into other vessels downstream that we can't see. That's why you're losing the color or the brightness. but it's not going into those organs like we saw on the previous slide. All right. So what does this mean? Okay, so the seal is a really good model to study this. So what's actually happening is, first of all, there's a selective vasoconstriction that's preventing the flow of blood into these, we call non-essential organs. When the seal's diving, doesn't need its digestive system, right? It doesn't need a lot of the other organs. It basically, most of the blood needs to be contained between the heart, the lungs, and the brain. So it shunts blood away from these organs and keeps it in the core, right? So what does that do? That obviously those are non-essential organs right now, but also it helps to keep the level of oxygen up. The oxygen is not being metabolized as quickly because it's not going to those organs. So it allows the oxygen to stick around longer, right? And combine that with the myoglobin. That's one of the organs that the blood does go to. The myoglobin is very high, so it helps to keep the oxygen in the muscle. Obviously, the seal needs the muscle for diving, right? But it doesn't need it to those other organs. That's one aspect. The other thing is that the heart rate decreases. And this all falls into play if you look at the big picture, right? Because we're not using up oxygen as quickly because of the vasoconstriction, right? Our metabolism is decreased. The heart can slow down, right? So we don't need to pump the heart as much. If you've ever done, I teach a physiology class at Mesa where we do this simulated diving reflex. It's kind of fun. We don't have a big tub or anything like that, but what I do is I have students, we have like a little, kind of a small little tub like with ice water, and I have students, one student in a group stick their head in that bucket, and they're hooked up to a pulse transducer that measures their heart rate. And within a matter of seconds, their heart rate goes from, I don't know, 70, 80, all the way down to 50. It just drops, all right? And that's a really good diving response. That's also why, if you've ever seen, you've heard about in the wintertime, there's always somebody that in a cold part of the world or east or skating on a frozen lake falls through the ice. They go chasing after the person. Hopefully they find them. And if they do find them, it might be after 20, 25 minutes, and you think, well, this person's not going to make it. Quite often they do because of this diving reflex. Their heart rate slows down, right? So they're not metabolizing because they aren't metabolizing as much oxygen. So oxygen is just more available. So it's pretty cool. All right. So stop share, and we still have a whopping 5 people here. All right. Well, you guys, thanks for showing up. I know everyone, it's a hard time for everybody as far as making it, but I really appreciate it. I hope you're getting something out of being here live as opposed to just, looking at the video whenever. Let me see, someone's asked a question. Thank you, thank you. Okay, someone said thank you. Okay. Oops. I lost it for a second. Okay. So anyway, are there any questions? All right, so I'll send this out to you guys shortly, and I will see you tomorrow. We'll start going over the respiratory system.